Here is a quick refresher for passive transport. First of all, one thing that I want you all to remember about passive transport, regardless whether it is diffusion, osmosis, or facilitated diffusion, is the fact that it does not require any energy from the cell. And that's a really big deal because all of that energy that isn't used for passive transport can then be used for other important processes that keep the cell going. Something else that you definitely need to remember about the process of passive transport is the fact that it's the movement of molecules or particles from areas where they are very highly concentrated out to areas or into areas where they are less concentrated. It doesn't literally mean they're moving from high to low, so it's not a directional movement of up and down, but rather where stuff is located, from where there's a lot to where there's a little. One way to kind of help you remember in a visual way the idea that there's no energy required and the fact that it's movement from high to low concentrations is to think about rolling a ball downhill. Rolling the ball downhill is easy because it does not require much energy and it's moving from high up to down low. Not an exact definition, but it's a visual that might help you to remember what passive transport is all about. Moving on. So what I'd like to talk about next is the basic movement into and out of a cell, how you can tell which way it's going to travel, and then also quickly recap the different types of solutions that exist: hypertonic, hypotonic, and isotonic. So in this diagram, we have the image of a cell. This is the cell membrane, and inside the cell we have two molecules, the blue and the green dots. The blue dots in this example might represent something like water molecules, and the green dots could represent sugar or glucose. As we know, diffusion is the movement from particles, diffusion is the movement of particles from areas where they are highly concentrated to areas where they are less concentrated. So, if you were a glucose molecule, one of these little green guys, which way would you be traveling? Would you move out of the cell? Or would you move into the cell from the outside? If you answered out of the cell, you are correct. In this example, there aren't even any of the glucose molecules on the outside, so high to low concentration is going to be from where there are some to where there is none. A fairly simple explanation, but it works. Conversely, if we take a look at water, and remember, what is the diffusion of water called? Osmosis. So, if you are a water molecule, the little blue dots, which way are you going to travel? Into or out of the cell? Remember, the same rules apply, so if diffusion is the movement of molecules from high to low concentration, osmosis is the movement of water molecules, again from high to low concentration. So the water would be moving into the cell, because there's a lot more water on the outside than there is on the inside. Pretty simple stuff. All of this is important to know because in life, everything is all about balance. Your cells need just the right amount of everything: oxygen, carbon dioxide, salt, waste products, water, sugar, vitamins and nutrients, etc., etc., etc. Some of these things your cells only want a little tiny bit of, and some of these things your cells want a whole lot of, but what they all have in common is movement into and out. And the main thing to remember for this particular slide of equilibrium is just that if there is an equal amount of particles on the outside and the inside of a cell, there might still be some movement out and there might still be some movement in, but there's really no net movement because you have equal parts on both sides. This is a good thing. This is generally where we want to be in all aspects of life. This leads to an idea of homeostasis, which we're going to talk about a little bit later. So last thing to recap are just the three types of solutions: hypertonic, hypotonic, and isotonic. The first one I want to go over is hypertonic. Now, really quickly, in case I didn't say it or in case I haven't said it before, regardless whether it's hyper, hypo, or iso, all of these are referring to the solution. And the solution is just the liquid part on the outside. Doesn't matter. In our diffusion lab, for instance, the other day, the solution was the combination of water and iodine that we had in the beaker. A solute, on the other hand, solute, is referring to whatever is dissolved into that solution. So in the diffusion lab, water was the solution and the solute was the iodine that we put into it. So with hypertonic, hypotonic, and isotonic, hyper from hypertonic, the Greek derivative of that root word means over or above. I like to think of that as basically just meaning a higher number. Over, above, big, more. All of those words are very similar in their meaning, and it helps you to remember what hypertonic is referring to, because hypertonic solutions have more of that solute, more of that dissolved substance, more of that particle in it compared to the cell. Not as much, more. Not as much, more. Not as much, more. So, in a hypertonic solution where there is more of that solute, the movement is going to be from the solution into the cell. Again, high to low concentration. You're just looking at the overall amount. You're only identifying it by calling it hypertonic instead of just counting the dots. On the other hand, if we look at this diagram, we can see that there's a lot more of that orange inside the cell, and there's only a few specks of it here and there outside of the cell. In this case, hypo, meaning under or below or less, is referring to the fact that in a hypotonic solution, there's less of that solute, there's less of that dissolved substance compared to the cell. And if movement is always going to be from high to low concentration, the picture is easy enough. In the picture, we can tell that movement will be out of the cell by these tiny orange dots. But even just by looking at the word hypotonic, you should know that that is going to mean that movement is going to be out of the cell of that particular substance. And finally, isotonic solutions. Isotonic or iso means the same. So we have the same amount of solutes on the outside as compared to the inside. Therefore, there might be a little bit of movement out and a little bit of movement in, but overall, there's going to be no net movement. There's not going to be anything. This is in a state of equilibrium, that word from earlier in this presentation. Finally, one last thing that I want to mention. In hypertonic, hypotonic, and isotonic solutions, if the solute, remember that's the dissolved substance, if the dissolved substance is trying to move into the cell, to make things equal, what's going to happen to the water, and I'm going to change colors just to emphasize that we're talking about the blue dots now. There we go. Water is going to be moving out at the same time that the solute, the dissolved substance, is moving in. So the net effect of this, and I'm going to clear the page for just a second. There we go. The net effect of that in a hypertonic solution is that water is actually going to be moving out of the cell, and whatever you've got dissolved substances of, maybe salt, maybe glucose, etc., the solute, right, the dissolved stuff, the solute is going to move in. That is in hypertonic. On the other hand, in a hypotonic solution, if the solute is moving out, which we discussed, then to keep things even and to fill in that space, water is going to rush in. I will clear this slide as well. Draw it again. Here's the cell. In hypotonic solutions, water is going to move into the cell, water, and the solute is going to move out of the cell. This will cause the cell to get bigger and sometimes explode. Isotonic, nothing happens, we're all good. Hypotonic, death by explosion. Hypertonic, if water is leaving, it's actually going to cause the cell to collapse. It gets all shriveled up. All right, there we go. If you should have any questions, please see me or rewatch this a couple of times, or use Google, or look up some YouTube videos, or do whatever you have to do to understand the process of passive transport.